Teaching Tips in AP Biology number 2. This video presents formative assessment activities for the enzyme lab. This activity is best done after completing the enzyme lab. It's modeled on enzyme catalysis on a shoestring. It's formative assessment if you use this activity to see what students know and help them improve where needed. This will involve discussions between students and with you as students refine their knowledge. The way this activity is structured, students will see they are working with science practices as well as content. I have made a pair of videos for this formative assessment. You could use them as a virtual lab as well, particularly for students who are absent. You can find them on YouTube and the URLs are here. I will be using them in this video, but when you use them with students, I would suggest you direct them to YouTube where they will have the full video and pause capability. This is the beginning of video 1, the control. The catalase is in a yeast solution. It's reacting with hydrogen peroxide to generate oxygen gas. About 1 milliliter of yeast solution is added to a small amount of hydrogen peroxide. Oxygen gas is collected through water displacement. The second video is the experimental condition. Protease has been added to the yeast solution and allowed to sit for 30 minutes. After this, the yeast solution containing catalase is added to the hydrogen peroxide as in the control. As students watch the two videos, you may want to have them with the questions in front of them so that they can consider their answers. I will look at each question individually and offer a sample answer. After watching the beginning of both videos, they will think about what questions the researcher may have been asking. There is more than one way to look at this, but here is a common predictable response. Does protease break down catalase and stop or reduce its action? The point is, can the students pose a scientific question? The next step is the formation of a hypothesis. The students should be able to take their scientific question and extend it by predicting results based on scientific principles. As students begin to form their hypotheses, look for connections between protein structure and function and the ability to catalyze the reaction. The next question asks students to describe the control and experimental groups and to predict what will happen in each of these two conditions. The prediction may first be verbal, but will also need to be graphical. The predicted graph shows two lines, one for the control without protease and one for the experimental group with protease. Require students to label this graph and you will be helping them practice skills they need for the AP Biology test. Let's look at the control video first, that with no protease. The students may need to watch the video several times to decide how they wish to collect the data. In this example, I've collected the data by waiting until the bubbles where the gas and water meet reach the 1 milliliter line. At that point, I start the stopwatch on my iPhone. Here is the first point. For the second mark, I hit the lap counter. Each time the bubble reaches a 1 milliliter mark, I hit the lap counter. This is repeated with the protease group. It is apparent to the naked eye that this reaction is much slower than the first. The real task is to make the case with their data. Will the data support their first impressions? Since I collected the data using the lap feature on my iPhone, I feel like I should explain to students how I translated it into an Excel graph. That is done here. With the lap counter data, I had to add each lap to the preceding lap to get cumulative time. Since time was my independent variable, I used the actual times for X in Excel. These were paired with the milliliters of gas at that time. Excel generated the equally intervaled x-axes from these different times. However students go about it, they need to be able to explain how they collected data to answer their question. I put the data in Excel and had Excel do the trend line and place the equation of the line on the graph. The skills needed for Excel are well explained in the video referenced on the previous slide. The student should calculate the rate by hand as I have shown here, and then compare it to the equation of the line. I would suggest you have students compare their calculations with the equation of the line and see how these two things interact. After that, the students will do some writing where they are justifying their claims with evidence and constructing explanations. In the next couple of slides, I give an example of how they might do that. Students that finish more quickly may want to make a rubric for grading this kind of question. They can present it to the class, and students could use it to evaluate their writing. It's really important that they practice the writing. In the last part of the video, I showed how to connect the evidence students saw with explanations of phenomena that tied in the structure of proteins and the actions of enzymes. I hope this was helpful in using formative assessment with students. Until next time, this is AP Biology Teaching Tips.